anything like that. But before you can have macromolecules, you have to have simple monomers, right? So we talked about the hypotheses that exist to explain how we got that far. There's going to be an FRQ when you have to talk about the evidence that exists that defends the different hypotheses associated with the origin of life. For example, we talked about the operant hypothesis, Haldane's hypothesis, reducing atmosphere, primordial soup. What evidence does support? We talked about panspermia. We talked about, what else we talked about? That is spontaneous formation of lipids. We talked about heated clay sperm. No, not talking about the spontaneous formation of polymers, protein polymers from heat sources. We talked about Miller-Urey experiments, and I showed you a video of this. Make sure you know that evidence. Now, the next step is just logic. All right? How do you go from those building blocks, which we could potentially, remember, all we need is a possibility, have available to actual life? For that, because I am short on time, I'm going to leave that to the end if I have time. But there's a video by Think About It that I'm going to send it to Sally if you don't have time, because we only have 15 minutes. All right? But the video is called The Origin of Life. Remember I told you to watch Think About It videos? Right? One of the videos is called The Origin of Genetic Code. I love that one. The other one is called The Origin of Sex. How could such a complex thing like sex have evolved? And more importantly, why did it evolve? Right? The other one that's very interesting is this one is called The Origin of Life. What that video will do is it will piece everything I'm about to say together for you. So this is how you connect the things. All right? So what we need to have evolution, to have a life form. Let's talk about characteristics of life. You have to have a self-replicating system that has homeostatic control, that exchanges materials with the environment, right? That has genetic information of some sort, right? That has focus on reproducing and passing that on to the next generation. Right? How do we get there? Well, here's the thing about those bubbles that we talked about. What happens to those bubbles that you form in water if you shake the water? What happens to that? Have you tried shaking water that has bubbles in it, or made of oil? What happens to the bubbles? They split up. Correct? Yeah. So as long as the water is moving around, any bubble that forms from fat. Remember how the bubbles form in fat in water. How do fat bubbles form in water? Interactions, right? It's just by physics. They will find each other and form together, right? But they also can break apart by the movement of the water. Now I have to imagine an early Earth with a current that would move the water around and make that happen. So now we have a system to make bubbles and a system to break bubbles apart into smaller chunks. Now here's the thing about these hydrophobic bubbles. All right? This is very important. Remember, don't write just what I write. Write the things I say. It's important. But it is being recorded, so if you were overwhelmed, right? But the thing about those bubbles is that the bigger they get, the more they attract other bubbles. So larger bubbles tend to grow faster than smaller bubbles do. But larger bubbles also tend to break easier than smaller bubbles do. So it's kind of a conundrum. But let's think about this. If I'm a tiny bubble, I will probably end up absorbed by a bigger bubble. And as the bubble gets bigger, the currents, they break it down and spread around in these branches, which makes it more likely for them to break apart. Right? So notice this is very important. This is the key for a bubble to get larger and get bigger. Anything that will make a bubble will give an advantage and make it replicate faster and divide faster. Are you with me? Something that can make bubbles grow faster. Osmosis. Now you learned this in biology, so hopefully you know this. What happens when a cell gets full of salt inside? What happens? Water should go inside. Osmosis, right? But what would that do with the size of the bubble? Okay, so if anything is inside, more water will rush in, which will make the bubble bigger. But if the bubble gets bigger, it will divide faster. So all I need to have an advantage over other bubbles is have something inside. Like, for example, a self-replicating molecule that's working on making copies of itself. And the more copies it makes of itself, the more salinity is inside, the more water goes inside, the more it tends to divide. So any cell with self-replicating molecules on the inside, any protobiota, so to speak. So what we need to really have this early protobiotic life is have an accident of a self-replicating molecule being created inside of a bubble. How would that happen? So you have fat layers. Now here's the thing about fat. If we do this at the end of the year, we're actually going to do this. To let something go inside of the cell, we can heat it, heat shock. So if we heat shock it, what happens is that the membrane expands really quick. As it expands really quick, small things can get inside. Are you with me? Things small, like an amino acid, produced by either coming from a comet or produced by a process similar to the Miller-Urey experiment. So now that amino acid is inside the bubble, right? One of them. Or maybe two, or maybe three, or maybe four. And that happened because it was heated. Heated by what? Maybe a hydrothermal vent, a volcanic eruption. Right? So near a volcano, bubbles are forming, and then this heat, as the bubbles pass through the volcano, which by the way is shaking it, making them split, which also helps with all this. But the heat makes the bubble expand, which allows amino acids, which are forming in the heated clay that's right next to the volcano, right? Get inside. Now, because it's convection now, it's physics. Hot water and bubbles tend to what? Rise. As they rise, they cool off. As they cool off, the membrane closed down, and now the things are trapped in the inside. Are you with me? But this thing is a current, so it makes a current like this, and eventually it'll go back to the hydrothermal vent again, right? They do like this loop, just like a boiling water, loops around. So eventually it'll go back to the hydrothermal vent, and the process repeats itself, and more amino acids get trapped on the inside. Some will leave, some will come in, but more of them will come in, will end up being trapped inside by simple diffusion. Why? Because as long as there's more amino acids outside than are inside, they will tend to hurt inside by simple diffusion, physical process. Are you with me? Then as soon as we have those things inside, the heat of the volcanic thermal vents, just like the heated lava clay, they make them polymerize spontaneously into peptide chains, or amino acid chains, or nucleic acid chains. Simple molecules, right? Now, these chains have no meaning. They have no function. We talked about this. They're just chains of peptides, or chains of nucleic acids. They have no meaning. So great. But no, what they're doing though. Because that molecule is in there, it is making the thing go faster, and therefore splits faster, right? When they split, some of the amino acids go with one of the bubbles, some of them go with the other. But they already have some inside, which means they gather more. It makes the process, it's differential selective reproductive success, although it's not technically alive yet. Well, at what point is it alive? Now, that sequence is insignificant. But any sequence that has amino acids, or let's say nucleotides, which are very common out there, that were made a lot, or that came a lot from volcanoes, will be advantageous. Because if you have that sequence, it will be more likely for you to replicate yourself. And that's how you end up with 4 codons, of the 4 ACTG of DNA. Because they were more common, and so early in the life process, they were selected for. Because any type of life that was trying to develop without those 4 basic ones would have trouble finding the building blocks to be copies of itself. And so they were selected for. But any sequence, even though it has no purpose right now, but any sequence that accelerates in any way the copy of replication of itself, either by having easier to find pieces, or by having any kind of property of being self-replicating, would be selected for because they would make the thing go faster, right? And from that, you kind of start sounding like a sequence that has a purpose to self-replicate itself. And now we have a purpose. Now it's no longer random. Now it is natural selection. And from that, the rest is history. Now, that is a very interesting thing. And to really understand it, you have to watch that video. I really think it would do a better job in this quick review that I did. But a few more things that I have to talk about. We have another hypothesis called the RNA world hypothesis, which says that the most likely candidate to be that first molecule was probably RNA. Why? Because RNA has multiple jobs in life today. Throughout the year, we're going to learn about this. They help synthesize protein. Some of them act like enzymes. They're called ribozymes. So they do the same thing that enzymes do. Other parts of RNA are structural, like the RNA that can be found inside ribosomes. Other parts of RNA are guides or chaperones or transport proteins, like hemoglobin is a transport protein. There's an RNA protein called the SRP molecule, which finds the ribosome and drags it to the RFER when a protein is going to be made to be exported from the cell. There's different types of RNA that do different functions. There's an RNA that's part of the spliceosome, which decides which parts of the DNA to cut before the protein is being made in eukaryotes. There's a multiple different, there's RNA that helps to regulate other RNA. RNA basically does, in different forms, different types of RNA do a lot of the different jobs that proteins do. So you can think of them as an intermediate. Also, you can easily see how an RNA doubled, caught around itself, could become a double helix. So it makes sense that the RNA would be the first, right? That it's simpler, and it could double. And it's also today the bridge between DNA and proteins. So in a simpler early life form, we think the RNA world hypothesis makes sense because of all these things I just said, right? That's another hypothesis associated with the origin of life. Now we have life. What are some major milestones that go from there? All right. First is the evolution of nitrogen fixation bacteria, which transforms an atmosphere of ammonia-rich, right? No, not fixation bacteria. Sorry, nitrifying bacteria, which transforms an ammonia-rich atmosphere to a nitrogen-rich atmosphere, right? Then we eventually have the evolution of cellular respiration, allowing us to break down sugars. Now, no longer we have to live close to hydrothermal vents, where production can be made with complicated chemical processes. We can store sugar and live far from the vent, or eat someone that makes sugar, right? We're talking about glycolysis, they break it down with sugar, right? After that, in a kind of reverse glycolysis, which is what it is, photosynthesis. The evolution of a type of life that can capture sunlight, cyanobacteria were the first one. Stromatolites are evidence of that. Stromatolites are evidence for that. What is the evidence that cellular... I skipped that, because every step we have to talk about evidence, right? What is the evidence that cell respiration was an early step? That glycolysis is such an early step? Because what? 99% of life on Earth does it. Why? Because it's such a conserved core process. It must be a very ancient one. By a core process, means something so important that every single life form has it. You with me what I'm saying? The genetic code. It's the same for everybody. What the DNA is built of. What RNA is built of. The 20 amino acids that build up proteins. Those are all conserved core structures. Right? The cell membrane. The basic structure of the protein synthesis machinery. That's a process, conserved core process. A conserved core process is a process that everybody does. A conserved core structure is something that everybody has. For example, among all life forms, you have the same building blocks for DNA, the same building blocks for RNA, the same structure for DNA, the same structure for RNA, the same building blocks for proteins, and things like that. Those are conserved core structures. Among eukaryotes, all eukaryotes, well, we're getting ahead of myself because we're not talking about eukaryotic evolution yet. Okay, so these are conserved core structures that I just mentioned. Then you have conserved core processes. Glycolysis, everybody does it. Photosynthesis, everybody has a similar one. DNA replication, everybody has a similar one. These are characteristics which are evidence of early unity in the history of life. Because everybody does the same way. Because we all come with a first original type of life form. Are you with me? So conserved core processes and structures show unity to all life. It's kind of something that's homologous to all of us. Are you with me what I'm trying to say? Yes? Okay. Then, after we have glycolysis and photosynthesis, now we can start making our own sugar with the sunlight. We're not trapped at the bottom of the ocean near volcanic vents anymore. We can go anywhere near the surface, perhaps even one day to the land. Photosynthesis, eventually, we also develop aerobic respiration because now there's oxygen in the air. And because there's oxygen in the air, we can do aerobic respiration. So after aerobic respiration evolves, the process of making energy out of sugar becomes 9 times more efficient. And now with more energy, life can explode and do a lot more. We get the Cambrian explosion. We have a lot of things that get evolved because of that. But even before that, evolution of eukaryotic life. Alright? That's much more complicated. I think I'm rushing too much. You know what? Next class, I'm going to spend 30 minutes to do this slow. Because I want to talk about these early milestones of the history of life. But there will be, however, on the second half of class, another FRQ. Alright? FRQ shall be on evidence for evolution. Alright? And something else I'll throw you as well. But there will be an evidence for evolution. Remember the chart with the evidence for evolution? Make sure you know those evidence for evolution and you'll be in good shape. Alright? Now, this is a chance for you to redeem yourself. For evolution, make sure you're ready for next time for that. So, next class we will revisit the early milestones of the history of life in the conserved core processes and core structures that allows us to say that life all started from one original life form. Right? Oh, actually, with the 30 seconds I have left, let me give you one last thought. Okay? One last thought. Okay? Listen to me. Evolution and biogenesis are two different things. If evolution is wrong, evolution can still be right. But here's the thing about evolution. Although it's not about how life started, it kind of implies that life started from an original life form. Because when you think about evolution and common descent, eventually the ultimate result of common descent, right, is that one origin that binds us all. Right? So that's the thing. We all descend from modification from something that came before. And we all have cousins and cousins and cousins and long distance more. Eventually, we have to talk about a root. And so it makes sense to talk about one origin from one original life form. Alright? So evolution and biogenesis are different things, but one is implied from the other.